So in this morning's session, the CLL Forum session, we were delighted to have both international speakers and our own homegrown talent. Jan Berger came over from the MD Anderson, Arnon Keita came over from Holland, and Anna Xu came up from Oxford to present data. It was a really great session, giving the real flavour for the excitement in the CLL world. Jan Berger, who has been at the forefront of many developments in CLL, both biology and therapeutics, was able to link these two aspects together, showing us the way forward in therapeutic trial design. Arnon Keita, great data, a lot of primary laboratory work and how that linked into their clinical trial work and the direction of travel for the HOVON group. And then Anna Xu was able to show us a lot of molecular profiling and really the direction of the future, where CLL cancer really is going to become a much more individualized, with personal medicine directed specifically at abnormalities wrong in the cancer, the specific cancer for that patient. Yeah, so the microenvironment is absolutely fascinating in CLL. We've known for years, if you take CLL cells out of the blood, put them in the lab, well, they simply die off. They need the microenvironment. They need the nurse cells, the macrophages, to actually look after the cell and give them growth signals. And what's become fascinating is understanding how that interplay works, what it's dependent on, and of course, from a pharmaceutical perspective, how it can be targeted. Because if you can kill off aspects of the microenvironment, well, you can kill off some of the helper signals that the cells require to live. And it's really, really exciting seeing that work coming through from Arnon and Jan, showing that a lot of these therapies we're using are targeting that environmental interaction. Probably one of the reasons why some of these new great drugs like ibrutinib are so effective. It's a very good question to ask about microenvironment variability. We tend to focus much more on cancer variability. So in CLL, the CLL splits down the line between the mutated immunoglobulin change and unmutated immunoglobulin change. They've actually got very different dependency on the microenvironment. Probably affects some of the differences we see in the responses with these new drugs. But actually the microenvironment itself, not very much is understood about how that varies between individuals in CLL. People are looking at the proteins that interact with the environment. Can these be targeted with new antibodies? Can you interfere with how the cells migrate through the tissues of the endothelium of the blood vessels? And there were a couple of presentations yesterday in the session I chaired from the lymphoid communications, a couple of great sessions from Cardiff, where they've been constructing artificial circulatory and microenvironment models, which are quite hard to imagine, but you've got to see the pictures to understand it, where they can mimic how cells move between the different compartments, between the blood into the lymph node, and how those interactions and trafficking can actually be targeted with new drugs. Well, I think it isn't yet quite ready for mainstream clinical practice. Undoubtedly, it's an area that will see much development in the future, but how cells interact with the microenvironment and cellular components such as, for instance, the T cells, the immune network, I haven't even mentioned PD-1 inhibition and these pathways, well, they've got great promise for the future. They're not quite there in the clinic just as we speak. We'll probably be talking about a completely new set of proteins and microstructures that we're not even aware of at the moment. But I think microenvironment targeted therapies will be coming our way, but perhaps more in the 5-year time frame.